Good day, my name is Fatma Bungkarawan and today I'm going to discuss about the current threads in cell and molecular biology part 1. Now, the current trends in cell and molecular biology include advancements in single-cell technologies, CRISPR-based genome editing for precise genetic modifications, and the growing use of artificial intelligence and machine learning for data analysis. Additionally, research in cellular senescence, immunotherapy, and the study of extracellular vesicles for intercellular communications are gaining prominence. These trends collectively contribute to a deeper understanding of cellular processes and hold potential for breakthroughs in medical applications and technology. Now, there are so many current trends in self-mold, but we're going to mention few of them or some of them. So without further ado, let's begin. Okay, so one of the famous trends in cell mold is CRISPR. Now, CRISPR, an acronym for Clustered Regularly Interspaced Short Palindromic Repeats, represents a revolutionary breakthrough in genetic engineering and molecular biology. Originating from the microbial immune system, CRISPR technology allows for precise modification of DNA within living organisms. At its core, CRISPR employs RNA molecules to guide molecular scissors, typically Cas proteins, to specific target sequences in the genetic code. This ability to precisely edit genes has transformative implications across diverse fields, from basic scientific research to potential therapeutic applications. CRISPR's ease of use, cost effectiveness, and scalability have catapulted it to the forefront of genetic manipulation tools, enabling scientists to modify genes with unprecedented accuracy and efficiency. The advent of CRISPR has ushered in a new era in genetic engineering, sparking innovations, controversies, and promising avenues for addressing genetic disorders and advancing our understanding of fundamental biological processes. Now, the evolution of CRISPR techniques has witnessed significant strides, particularly in enhancing precision and minimizing off-target effects, initially centered around the Cas9 nuclease. And before that, let's give an insight of what is Cas proteins. Cas proteins or CRISPR-associated proteins are essential components of the CRISPR-Cas system and a revolutionary gene editing tool derived from the microbial immune system. These proteins work in conjunction with guide RNA to precisely target and modify specific sequences within an organism's DNA. The most well-known Cas proteins is Cas9, but others like Cas12 and Cas13 also play distinct roles. Cas9, for instance, acts as a molecular pair of scissors, creating double-stranded breaks at targeted DNA sequences. CRISPR technology has evolved to incorporate various Cas proteins with unique properties such as Cas12 and Cas13. Innovations like base editing and prime editing allow for more precise modifications at the nucleotide level, reducing the reliance on inducing double-strand breaks. The design of guide RNAs has been optimized through advanced algorithms, contributing to improved target recognition and specificity. Structural insights into Cas proteins have enabled engineering for higher fidelity, ensuring accurate binding to intended DNA sequences. Additionally, machine learning algorithms assist in predicting potential off-target sites, and optimized delivery systems enhance the effective targeting of CRISPR tools within cells. These advancements collectively contribute to the increased precision of CRISPR techniques, making them more versatile and reliable for applications in genetic research and potential therapeutic interventions. Now, how does CRISPR basically work? So, Here is the image shown. Okay. Okay, so here are the steps. The first step is that the Cas9 protein forms a complex with guide RNA in a cell. The guide RNA is a crucial component in the CRISPR-Cas9 gene editing system. It is a short RNA sequence that is designed to be complementary to a specific target DNA sequence. In the context of CRISPR gene editing, the guide RNA serves as a guide to direct the Cas9 enzyme to the precise location in the genome that scientists want to modify. The second step is that this complex attaches to a matching genomic DNA sequence adjacent to a spacer or in the yellow segment. The third step is that the Cas9 RNA complex cuts the double strands of the DNA, and we mentioned a while ago that the function of Cas9 protein is a molecular scissors that cuts the DNA sequence. The double strands of the DNA. And the fourth step is that the programmed DNA may be inserted into the cut. So here is one of the application of CRISPR gene editing tool to some diseases. So, for example, is a sickle cell anemia. The sickle cell anemia is a genetic blood disorder characterized by the presence of abnormal hemoglobin, the protein responsible for carrying oxygen in red blood cells. This disorder is caused by a mutation in the hemoglobin subunit beta gene, which provides instructions for producing a component of hemoglobin known as beta globin. The mutated gene leads to the production of hemoglobin S, resulting in misshapen red blood cells. The CRISPR-based sickle cell gene therapy approach focuses on repairing the mutation in the adult hemoglobin gene associated with sickle cell disease. In this method, Daniel Dever, a research instructor at Stanford University, introduces a DNA break to the beta globin gene using CRISPR technology. This break serves as a targeted point for correction through homology-directed repair, or HDR. The HDR process involves introducing a donor template containing the normal sequence of the gene. When the cell undergoes repair at the site of the DNA break, it utilizes this donor template, correcting the mutation in the process. This specific correction method is referred to as a gene knock-in. The resulting edited cells are then reintroduced into the patient's bloodstream, now engineered to produce healthy normal hemoglobin. What is particularly promising about Dr. Dever's work is the efficiency and reproducibility of the gene editing method, which allows for the mutation and analysis of precise genomic locations within a matter of weeks. Having completed preclinical development, the next crucial step involves moving towards clinical trials, marking a significant stride toward the potential implementation of this innovative and precise treatment for sickle cell disease. Okay, so aside from CRISPR technology advancements, one of the current trends in cell mold is the personalized medicine. Now, the personalized medicine, also known as precision medicine, is an innovative healthcare approach that tailors medical treatments to the individual characteristics of each patient. It involves considering the patient's genetic makeup, molecular profile, lifestyle, and environmental factors to make more informed and targeted decisions about disease prevention, diagnosis, and treatment. The core idea is to move away from a one-size-fits-all model and instead customize healthcare strategies based on the unique attributes of each individual. Traditional medicine often employs one-size-fits-all treatment strategies where interventions are designed based on average responses within a population. This approach may overlook individual variations in genetic makeup, disease susceptibility, and responses to treatments. Personalized medicine, on the other hand, recognizes and leverages the inherent diversity among individuals. It takes into account the genetic and molecular differences that contribute to variations in disease risk, progression, and treatment responses. Now let's talk about genomic medicine. Genomic medicine involves the use of information from an individual's genome to guide medical decision making. This field integrates genomic data, including DNA sequences, variations, and gene expression patterns, with clinical information to understand the genetic basis of diseases, predict individual disease risks, and tailor medical care based on a person's genetic makeup. And the advances in genomics have significantly contributed to the paradigm of personalized medicine by identifying genetic variations that inform healthcare decisions tailored to individual patients. So here are key aspects of how genomics contributes to personalized medicine. Okay, first aspect is pharmacogenomics. Now, genomic information is used to identify variations in genes involved in drug metabolism. Pharmacogenomic testing helps predict an individual's response to certain medications, involving dosage adjustments and avoiding adverse reactions. For example, is the CYP2D6 gene and codeine metabolism. Codeine is a prodrug that requires bioactivation by a liver enzyme called CYP2D6 to form morphine. The amount of codeine converted into morphine by CYP2D6 varies from person to person. A patient's CYP2D6 metabolizer status can be predicted from genetic analysis of the CYP2D6 gene. This information can inform the prescribing of the most suitable analgesic for that patient. Some patients with near normal enzyme activity are classified as intermediate metabolizers. If a patient's CYP2D6 metabolizer status is known, there are international guidelines that can inform the choice and dose of analgesic for that patient to improve the effectiveness and safety. These guidelines are summarized in the image shown on the right side. CYP2D6 is a key enzyme in codeine metabolism. However, there are many other genes involved in the absorption, digestion, and metabolism and excretion of analgesics. Okay, so another aspect is the hereditary disease risk assessment. So, for example, is the BRCA1 and BRCA2 testing. The BRCA1 and BRCA2 are human genes that produce proteins responsible for suppressing the growth of tumors. Mutations in these genes, particularly when inherited, are associated with an increased risk of breast and ovarian cancers. Genetic testing for BRCA1 and BRCA2 mutations assesses the risk of hereditary breast cancer and ovarian cancers. Positive results may lead to preventive measures such as enhanced screening or prophylactic surgeries. For example, is Angelina Jolie's decision to undergo a preventive mastectomy after testing positive for a BRCA1 mutation exemplifies how genetic information can inform proactive health decisions. Advances in genomics empower. This shows how advances in genomics empower personalized medicine by identifying genetic variations that influence disease risk, drug responses, and treatment decisions. This precision approach has transformative implications across various medical fields, guiding interventions tailored to the unique genetic makeup of each individual. Okay, so let's talk about biomarkers under personalized medicine. Now, biomarkers are measurable indicators that can be objectively assessed and evaluated as signs of normal biological processes, pathological processes, or responses to therapeutic interventions. In the context of disease and treatment, biomarkers play a crucial role in providing information about the presence, progression, or response to a specific treatment. Here is how biomarkers contribute to targeted therapies and the reduction of adverse effects. In predicting treatment response, biomarkers can predict how an individual is likely to respond to a particular treatment. This is particularly relevant in cancer treatment where genomic or protein biomarkers help identify the most effective therapies. For example, the human epidermal growth factor receptor 2 or HER2 is a protein which helps cells grow and divide. The HER2 gene produces HER2 proteins, and these are found as receptors on the surface of some breast cancer cells. Some people with breast cancer have higher than normal levels of HER2, and these are known as HER2 positive breast cancers. Approximately 1 in 5 breast cancers are HER2 positive. Tumors are tested for each HER2 as they are for the estrogen and progesterone receptors. There are two methods commonly used for testing HER2. First is immunohistochemistry and in situ hybridization or ISH testing. IHC testing or the immunohistochemistry testing is done routinely and determines how much of the HER2 protein is within the cancer cell. Results are either 1 plus, which indicates negative, or 2 plus, which indicates equivocal, or 3 plus, which indicates positive. ISH testing is done on request for all patients and is a confirmatory test which determines if there is an excess of the HER2 gene within the cancer cell. Results are either positive or negative. Now, the expression of HER2 protein in breast cancer is a biomarker or bioindicator that indicates potential responsiveness to HER2 targeted therapies like trastuzumab or Herceptin. So that's how important biomarkers are. So that's all for the current trends. I hope we learned more about the CRISPR technology advancements and the personalized medicine. So that is a few of some of the current trends and in Selmo. So I hope you learned a lot and thank you for watching.